Yeah, so thanks for that wonderful introduction. And thanks for having me. I'm really honored to be the first speaker in the seminar series, which I think is a great idea. Good job, Will. It's fun to not necessarily have to invite people to our departments to get to hear what people are up to. Especially, I'm not sure about your departments, but some of us are struggling with, paying people to come to give seminars and paying for travel and stuff, given the current funding climate. So I think this is an awesome idea. And I'm excited that this is a climate change focused seminar. I have spent the last like 4 or 5 years working on birds and not doing any climate change stuff. So I'm going to mostly talk about old research and then some stuff that my lab is doing now or will starting to be work on very soon. Um, now that I just started my position at Rutgers this semester, and uh, now have the opportunity to go back to some of these ideas that I'm going to talk about now. All right, so as I'm sure many of you know, emerging infectious diseases have been on the rise in recent decades, increasing risk to livestock and humans while contributing to catastrophic wildlife declines. And I think people generally have become more aware of infectious diseases than ever after COVID. One issue in particular that you'd hear a lot on the news, journalists, scientists, healthcare workers talking about and thinking about was why some people experienced mild illness while others were very sick, hospitalized, or even died. And another pathogen in the news all the time now is highly pathogenic avian influenza. It's another emerging infectious disease. And again, we have the same issue where like some birds are seemingly unaffected while others experience mass mortality events, like this Caspian tern breeding colony in Wisconsin where over 1,200 birds died. And understanding the sources of variation in disease risk and susceptibility is really key for understanding disease dynamics. And disease risk is not the same across species, populations, or even individuals. And to help us make more informed predictions and conservation interventions, we need to understand like what's driving these differences. So within a population, there are many factors that can create heterogeneity among individuals. You can imagine each of these toads has some trait like body size or diet that's represented by these different colors. Uh, and those traits might result in differing susceptibility to disease or disease severity that can help us understand trends within a population and make predictions about population level disease risk to better understand why some populations might be more susceptible than others. So this understanding of host traits and how host traits drive wildlife disease severity is a major focus in my lab. And I approach this from a number of angles, including thermal biology and climate change, which I'll talk about today. My research uses lab and field techniques and quantitative techniques to answer these questions across multiple organizational levels. And I usually primarily work in birds and amphibians. The last 5 years, I've been working exclusively in birds, and now I'm coming back to some of that amphibian stuff, which is what I'm going to talk about today. So this talk will be focused on amphibian systems and understanding how host traits drive wildlife disease severity in the context of climate change. So as you might know, temperature variability is increasing with climate change. And because of this, we need to understand how various ecological processes are affected by this variability. I personally have had some interesting experiences with these extreme weather events that I have here. So I grew up in South Florida, did my undergrad and my PhD in South Florida, lived there for the first 28 years of my life before I moved to Wisconsin. I moved to Wisconsin in January, which is an interesting time for someone from South Florida where it never gets below freezing to move to Wisconsin. And shortly after I moved there, there was a polar vortex and it was like negative 45 degrees outside my house. Really extreme event. And a few years later, I moved to Wisconsin, or not to Wisconsin, to Arkansas. And I also moved in the winter, thinking like mild winter, I'll be in Arkansas. Immediately, a polar vortex happens again. It's like negative 20 degrees outside in Arkansas. Just really shocking. The birds were doing all kinds of weird stuff outside our house and all these mass mortality events. And then I moved this summer to New Jersey, where within a few weeks of moving here, we had a really intense heat wave. And I went into the city to go to a baseball game. And when we tried to get on the train, the New Jersey Transit train, half the cars had no air conditioning. And it was over 100 degrees. I think it was like 102 that day. And everyone was jamming into these cars that had air conditioning. The couple that did, it was just like a miserable train ride. And then we got to the city and we're staying on the subway platform underground, no breeze. It's 100 degrees down there, extremely high humidity, just absolutely miserable experience. So we're experiencing these things now, and our infrastructure is obviously not prepared for it. And potentially wildlife aren't either. So again, one important ecological process that's influenced by temperature and by these variable conditions is disease. But unfortunately, the impact of temperature and variable climatic conditions on disease can be really difficult to predict. And the responses of host pathogen systems to weather are extremely variable, with multiple studies, even within single systems, finding that outbreaks occur more often at both warm and cold temperatures. And it might be straightforward to think about the relationships between temperature and a host or temperature and a parasite in isolation. Disease is an interaction between a host and a parasite. It's a complicated species interaction that makes things more difficult to predict. For example, we wouldn't expect this wood frog freezing over the winter from a really different climate than the squirrel tree frog in South Florida to respond to high temperatures in the same way. So host pathogen relationships are likely to vary depending upon the ecology of the host. My colleagues and I developed the thermal mismatch hypothesis to better explain this variation and how host susceptibility to disease is impacted by temperature. Those colleagues being Jeremy Cohen and Jason Rohr. And our hypothesis proposes that pathogens are able to outperform hosts under abnormal thermal conditions because pathogens have wider thermal breadths than their hosts do. So you can see that our hypothetical host, which is the frog in the blue line, has a more narrow range of temperatures over which it can perform well when compared to the hypothetical pathogen in red. And this happens because pathogens are smaller bodied and have shorter generation times than their host. And that allows them to adapt and or acclimate faster to variable climatic conditions than their host can. So because of these differences in performance, disease risk occurs at temperatures where the parasite outperforms the host, which is the circled arrows on the screen here. So that means cold adapted hosts will have the greatest risk at relatively warm temperatures, and warm adapted hosts the greatest risk at relatively cool temperatures. We'll see if I continue to say that straight through this entire talk, which under climate change conditions creates a rather dire situation for the cold adapted hosts, so you can imagine. So we can simplify the predicted impact of temperature on disease risk within non-extreme temperatures, which are these red boxes here, as a linear relationship. So again, to simplify, we have our cold adapted host on the left, warm adapted host on the right. When we expect our cold adapted host to have the greatest disease risk at relatively warm temperatures, and our warm adapted host to experience the greatest disease risk at relatively cool temperatures. So we primarily studied this in chytridiomycosis. It's a fungal pathogen or disease that's caused by two different species, Batrachochytrium dendrobatidis and Batrachochytrium salamandrivans, BD and B-sal. I'm just going to talk about BD today. They consume keratin in adult skin and larval mouth parts. And BD has been implicated in hundreds of amphibian extinctions and declines. It's widely distributed. And again, it's contributed to declines in over 500 species of amphibians. So that makes it a bigger threat to biodiversity than cats and rats, and the most harmful invasive species on the planet. And the growth of BD in culture has been well studied numerous times, many strains. Generally, it grows best in culture between 16 to 20 degrees Celsius and can be cleared on hosts for for some hosts anyway, can be cleared if you hold them above 28C for a few days or weeks. So the idea here is to create a framework for understanding how variation in host pathogen responses to temperature. So first I'm going to talk about predicting individual level variation, and then scaling up to think about predicted population level variation, and then predicting variation across multiple host pathogen species. And I'm going to try to end my talk on a positive note and talk about the conservation applications of this research. So first, let's talk about predicting individual variation and susceptibility with the thermal mismatch hypothesis. So within a population, individuals can vary in their preferred microclimates. So that's what these toads are down here. You have this one over here prefers a warm temperature. This one prefers a cold temperature. Within a population, there's lots of variability in where individuals are hanging out and the microclimates that they're using. So to determine those individual level temperature preferences, we individually place amphibians into thermal gradients. These are gradients I designed. They're not especially fancy. They're downspout gutters that I sawed in half long ways and then like coated with duct tape, essentially. There's a lot of duct tape involved, as you can see, but they work. We non-invasively measure their body temperature before and after BD exposure, giving us baseline and a post-exposure in individual level temperature preference. We did this for a handful of species, but for this study, I'm only going to talk about two, our coldest preferring species, the Atelopus ateki, and our warmest preferring species, the southern toad. And I'm going to keep visiting this prediction slide throughout the talk. So for our cold preferring species, we expect highest BD growth rates at relatively warm temperatures. And for our warm preferring species, we expect the highest BD growth rates in individuals that prefer cooler microhabitats. And that is exactly what we find. So each of these points is a single animal. And on the x-axis, we have their average temperature preference, so the microhabitat that they're choosing, the microclimate they're choosing, and then BD growth on the y-axis. So this individual level variation in thermal preference has real consequences for these individuals. And around the same time, we ran another lab experiment where we again determined thermal preferences using these same gutters, thermal gradient chambers, if I want to sound fancy, where we again determine temperature preferences for cold and warm adapted species. And this time we're adding in a species. This over here is a Cuban tree frog. And then we have our two from before. And so we have, the two warm adapted species, the southern toad and the Cuban tree frog preferring warm temperatures, and the Atelopus again preferring cold temperatures. We then acclimated all those animals to fixed temperatures within a range from 10 to 28C, infected them with their local strain of BD, and measured pathogen growth on both the host and in culture at those 6 temperature treatments. And what we find is that the two BD strains had similar growth curves in culture, despite coming from different climates. So we still have peak growth occurring around 18C for both BD strains in culture. But again, that's not what we expect to see on the host. So in the hosts, we expect BD growth rates to be highest for the Atelopus at relatively warm temperatures, and for our two warm preferring species to be highest at relatively cool temperatures. And that is exactly what we found. So for Atelopus, peak growth on the host was around 25 degrees Celsius. And for the two warm preferring species, peak growth on the host was between 10 and 18 degrees Celsius. So what we're not seeing here is a regression to the mean where, peak growth is happening at the same temperature and it's just relative in a direction based on what these animals prefer. We're seeing different peaks that can be predicted by the host physiology. So when we place that individual variation in microhabitat choices, in the context of host physiology, the direction of the effect can be predicted by the species level temperature preference. So talked about individuals. Now we're going to scale up and talk about predicting population level variation within the context of the thermal mismatch hypothesis. So to do this, to broadly test if thermal mismatches impact BD-induced mortality, I conducted a meta-analysis of BD lab studies that reported BD-exposed group paired with an unexposed control, reported sample size and mortality, and used wild collected individuals. And that last part is really important because we attached to this data set the climates that the host is adapted to. So using coordinates from where the individuals were collected, we got the 50-year mean temperature to use as a proxy for their adapted climate. And this resulted in a database of 206 data series from 58 papers, 47 amphibian species, 45 unique BD isolates. You can see there's some gaps here in the data. So the red are the isolates and the blue are the hosts. So we have nice overlap there, but we're missing a lot of studies from Asia and Africa. We just didn't find any. And then we had to get rid of some studies that used captive breeding animals from Australia and Central and South America, which are likely due to those species being imperiled and just not being practical to work with wild collected species or individuals. What we did have, though, was a really nice spread in lab temperatures that these experiments were conducted at. So to test the thermal mismatch hypothesis using this data set, incorporated an interaction between the host-adapted climate and the temperature at which the experiment was conducted. Again, this prediction slide with the expectation that mortality for cold-adapted hosts will be highest at warm temperatures and mortality for warm-adapted hosts will be highest at relatively cool temperatures. And that is exactly what we find. So across all of these hundreds of data series, this like diverse set of amphibian taxa, we find that for cold adapted hosts, they have the highest mortality at relatively warm temperatures, and warm adapted hosts have the highest mortality at relatively cool temperatures. And what was interesting is that most of the studies that I read through did not justify why they chose their experimental temperature. And the most commonly chosen experimental temperature was the temperature which BD grows best at in culture. And there's reasons why it might be useful to choose the temperature that the parasite grows best in culture for your experiment. But I think in general, these experimental temperatures should really be carefully considered, especially when you're designing experiments with conservation and management implications, which a lot of BD studies are. And working with ectotherms, which are particularly sensitive to temperature. So you might be thinking that I've just showed you a lot of lab studies. The first two were of the same species, and that's true. So now I'm going to talk about testing this in the wild using field databases. So we amassed a very large database of BD prevalence surveys. We had more than 4,000 amphibian populations from more than 300 papers. And again, we're attaching to this the climate and weather data for the purpose of incorporating this interaction between the host adapted climate, which is the 50-year mean, and the weather during the survey event, which is the temperature during the survey event. Pardon me. Yeah, so again, same prediction slide. On the left, we have cold adapted hosts, and we expect the highest prevalence for those hosts at relatively warm temperatures and the highest prevalence for warm adapted hosts at relatively cool temperatures. And I'm going to show these figures as curves instead of straight lines. But same idea. We're on the left going to have a cold-adapted species, which are species with mean climates below 15. And then our warm-adapted species on the right, which are mean climates above 20 degrees. And yeah, we find that these predictions hold true in the field data. The cold-adapted populations had a peak BD prevalence at 23 degrees Celsius, and the warm-adapted populations experienced peak prevalence at 16 degrees Celsius. So two different peaks, no regression of the mean happening here, or to the mean. These can be predicted based on the population's adapted climates, so the host thermal biology. So to put this another way, we find a negative relationship between climate and the temperature of greatest BD prevalence, where hosts at cool climates are at risk at relatively warm temps, and hosts at warmer climates are at risk at relatively cool temperatures. So let's think about how we can adjust our predictions here and how much these stand up to other conditions. So here I'm specifically going to talk about elevation and latitude. In this paper, we tested a bunch of different host traits, but I'm just going to focus on these two for the purpose of this talk. So how do we expect host population elevation and latitude to affect our predictions? Populations at high latitudes or high elevations and low latitudes should be more sensitive to BD under thermal mismatch conditions. And this is because those populations are experiencing lower interannual and seasonal thermal variation. So you expect them to have more narrow thermal performance breadths. So this first figure is for elevation. So it's going to look similar to all the other ones I've been showing. On the left is cold adapted hosts. On the right, we have warm adapted hosts. The solid lines are low elevation populations, and the dashed lines are the high elevation populations. And what we find is that at high elevations, amphibians from cooler climates are experiencing higher BD prevalence under warm conditions, while those at warmer climates experience high prevalence when conditions are relatively cool. But for low elevation populations, we show no impact of thermal mismatch on BD prevalence. So what we expect. Now, same thing with latitude. Here on the left is cold adapted host, on the right, warm adapted host. The solid line is low latitude populations. The dashed line is high latitude populations. And what we find is that at lower latitudes, amphibians from typically cooler climates are experiencing high BD prevalence under warm conditions, while those from warmer climates experience high prevalence when conditions are cool. While populations from high latitudes show a reduced impact, it's the same pattern, but a reduced impact of thermal mismatches on their BD prevalence. So, talked about individuals, talked about populations. Now I'm going to talk about making predictions across multiple host pathogen systems. So to do this, make predictions in variation across systems with the thermal mismatch hypothesis, we amassed a large database of disease prevalence surveys of wildlife populations that had specific dates and locations. For the same reasons that I described previously with the meta-analysis, we need to know exactly where they are. Because to this, we attach the 50-year climate data and the temperature during the survey event, so the weather data. And this resulted in a really large database. So we had more than 7,000 population level records and more than 2,000 unique host pathogen combinations, which are all mapped out here. We had much better global coverage with this one than with the meta-analysis. So again, this is just the same interaction I've been showing over and over again. It's climate interacting with weather. And that's how we are testing for the thermal mismatch hypothesis. And again, same prediction slide. So for our cold adapted species, our populations, we should expect highest prevalence at warm temperatures. And for our warm adapted species, we expect higher prevalence at cold temperatures. And again, it's interesting to think about this in context of what traits can alter these predictions. So here, again, we test quite a few of these in the paper. I'm only going to cover a couple, but first I'm going to talk about thermy. So we expect ectotherms to be more sensitive to pathogens during thermal mismatch conditions because they cannot regulate their body temperature independently from the environment when compared to endotherms, which should be less sensitive. So that's what this figure is going to show. So on the left is, again, cold adapted host. On the right is warm adapted host. The green lines are ectothermic, and the purple lines are endothermic hosts. And so what we find is that the thermal mismatch hypothesis predicts disease prevalence across ectotherms, but not endotherms. Endotherms tend to experience the greatest parasite prevalence during cold weather, regardless of the host adapted climate. And this could be an effect of cold-induced physiological changes in endotherms that can be immunosuppressive or increasing pathogen transmission rates due to behavioral changes that happen in the winter, like flocking, or other aspects of winter that leads to increased airborne pathogen transmission. So I've talked a lot about hosts, talk a little bit about parasites. So how do parasite types change our predictions? So we categorized all of the parasites in the database and tested how different taxa responded. So we split into bacteria, fungi, helminths, and viruses. So that's what I'm going to show here. So on the left is our cold adapted host again, and on the right is our warm adapted host. And then we have our four taxa, parasite taxa, split into different colors. And what we find is it's complicated. Fungal prevalence exhibit the strongest thermal mismatch effect, increasing sharply under cold conditions and warm climates, but more gradually under warm abnormalities and cool climates. By contrast, the helminth prevalence was positively related to temperature across climates, though this relationship was much stronger in cold climates, suggesting that warming may benefit helminths more than other parasites. Bacterial prevalence decreased under moderately cool temperatures in warm climates, but only slightly under warm temperatures in cool climates. And viral parasite prevalence increased under warm temperatures in cool climates, but not cool temperatures in warm climates. You see that's the purple line here. It's kind of flat. So we use this parasite type model to project change in parasite prevalence under various climate scenarios. The one I have pictured here is the representative concentration pathway of 8.5, which assumes little to no global climate policy. And continued heavy reliance on fossil fuels, which is maybe not all that unrealistic. And we can see those helminth and virus projections are really striking. So those are the two down here at the bottom, with far more increases than decreases, where the fungal and bacterial prevalence changes, those kind of balance out a little bit more. But regardless, this risk is expected to increase at higher latitudes, and particularly in the northern hemisphere, across all four taxa. So, talked about host traits and how they affect disease severity across individuals, and how we can use that information to make predictions at the population level. And if there is only one thing today that you take away from my talk, I hope it is this, that an understanding of wildlife and parasite thermal biology is really critical for predicting and mitigating disease, particularly in our warming world. So, as promised, I will now end my talk on a more positive study. Maybe this is a newer one that got a lot of press. Some of you guys have heard about it already, but talk about it anyway. So the work at the beginning of this talk that I spoke about with the individual level temperature preference stuff led to this really fun collaboration with Anthony Waddell and Rick Shine. And they wanted to apply some of these ideas to conservation strategies in green and golden bell frogs, which is this lovely frog here in the middle. So Anthony had some meetings with me, and he reconstructed my thermal gradients in Australia to use in his experiment. So he built the same, these are his, these are not mine. They have far less duct tape on them than mine, maybe a little nicer. So he reconstructed those and put the frogs in them to determine their temperature preferences before exposure. And then he exposed the animals, and individuals that were held at their preferred temperatures in incubators after infection, the orange line here, did better, and then ones held at a low temperature up here. And the ones that were allowed to thermoregulate freely did the best. So that's the blue line here. So those individuals fared much better than this control line, which are individuals that were held at a low temperature that's ideal for BD growth. So what they wanted to do was manipulate that behavior to help these green and golden bell frogs reduce their BD loads. So like I was talking about before, there's just all this individual level variation here in how amphibians choose their their microclimates and make decisions about thermoregulatory behavior. So really, we're taking advantage of that by increasing the availability of warm microclimates in the environment. So it's a really simple design that they came up with. There are these bricks, painted black, they have little holes in them. If you work with frogs or ever like look into a PVC pipe that's been hammered into the ground anywhere, there's probably a frog in it. They just really like hanging out in these little holes. I used to, when I worked with Cuban tree frogs, would just go out to the botanical garden on campus and hammer PVC pipes into the ground, and then just like come back later the next day and they would be filled with tree frogs, and I would just plunge them out and bring them back with me. A very easy way to collect tree frogs. And yeah, so we're taking advantage of this behavior that they already have. We're taking advantage of the fact that they prefer warmer microclimates and just creating the opportunity for them to use them. So bricks are painted black. They have these little greenhouses put over them that make them even warmer. And these sunwarm shelters, the control here is the same exact shelter, but shaded, so they're not being warmed. The sunwarm shelters increase the frog's body temperatures, which is great. That's what we want. And that increase in body temperature results in a decrease in infection intensity, which is also awesome. The lower their loads are, the more likely they are to survive their infections. And individuals who survive their infections and recover via heat treatment were more resistant to future infections. So these frogs are having warmer body temperatures by using these frog saunas, as someone so brilliantly decided to call them. Not me. I cannot take credit for that at all. They're warming themselves up. That warming is helping them reduce their BD loads, which helps them survive their infections. And when they survive their infections, they acquire immunity and are able to reduce their loads even more the next time they get infected. So it's a really awesome, very simple design, really awesome idea and implication of these research ideas, sort of more basic research ideas that I talked about previously in this talk. And if you want to make your own frog sauna, you can. This obviously doesn't work for every species, but it works well for the green and golden bell frogs. This is a QR code. Anthony wrote up a whole thing about how to make it and made it publicly available. They're very simple and very affordable. They're also the frogs are really cute, sticking out little holes. So Currently, my lab at Rutgers is interested in exploring the organismal drivers of the thermal mismatch hypothesis. So I have this sort of hypothetical picture down here at the bottom, where on the right we have a thermal performance curve, and this gray area is the range of temperatures over which the host performs well. When you introduce a pathogen, you can imagine that that range is narrowed. So I want to understand, we're working on understanding like what are the mechanisms there? And then how can we use that information, that organismal level information, to make better predictions for the future? And if any of that sounds interesting to you, I am recruiting a PhD student to start next fall. Applications are due December 1st. So send me an email, check out my website. If you're a student and you're looking for a PhD program, let me know and you think frogs are fun. And with that, I'll thank my co-authors and funding sources and frog sources and data sources and all kinds of stuff that's on here. That's it. A little early, but
Oh, that was perfect, Erin. Thank you so much. I felt
Mhm.
like I had questions all along the way, and then you ended up answering them as I got in there. I was like, oh, yeah, but what about the population level? Oh, tick that box. Okay, well, what about the
Perfect.
non-environmentally transmitted ones that don't have an environmental form? Oh, okay, tick that box. But so let's open it up for questions. Feel free to come on and ask questions of Erin. Or you can put it in the chat if you're shy, feel free. Let's see here. Well, while people are mustering up the courage to say something. Oh, did we have somebody? Oh, there we go. Go ahead.
Hi, Erin. That
Hi.
was a great talk. I'm wondering about the frog saunas and whether you ever have the possibility of transmission from frog to frog happening there, either because in that picture you had two frogs sitting next to one another, or just if they leave behind any pathogen that might get picked up by the next frog that uses the sauna.
Yeah, there certainly could be like contact transmission happening. I don't know that I don't so I am not in Australia. I cannot take like too much credit for designing these or like running that experiment. But I don't think it can grow or hang out on the bricks for very long. They are quite warm. And without a host there, Kitchard's not it's not going to live on the brick for any substantial amount of time. But they could definitely, if they're both sitting on top of each other like that, it could definitely be some direct transmission. But I'm not sure that the risk of that happening is any greater than the risk of them directly contacting each other anywhere else in space that they're hanging out. If anything, I think increasing the amount of microhabitats that are available to them could spread them out more and give them more opportunities to use these preferred microclimates, where without putting them in the environment, then they would have fewer microclimates to choose from in the first place. So I'm not, it could, I mean, there definitely could be transmission happening in the bricks. But I'm not sure in the grand scheme of things if it's any more or less than what's already happening in the wild. Yeah.
I had a question, Erin, about you showed in the last section, particularly like a lot of individual level variation in the types of temperature profiles that individuals themselves are seeking. And then also that like individuals who have the ability to kind of behave plastically in response to, their Kitrid challenge are better able to weather it themselves. So like as you're moving forward and thinking about these kind of individual drivers of the mismatch hypothesis, like do you think it's that like there's immune variation that's then, correlated with how these individuals are selecting the abiotic environments? Or like what's the driver, I guess, of that individual level heterogeneity in what they want to begin with and why that's so correlated with fitness outcomes?
I think there are lots and lots of things that are probably contributing to individual level temperature preferences. Some of that could be related to maternal effects. So if your mom laid your clutch of eggs in a pond that's warmer, like maybe you prefer warmer microhabitats. I don't know. That would be an interesting thing for somebody to look at. Uh, or I would think in the wild, there's probably a lot of competition driving that. I know that's true with lizards. There's a lot of competition for ideal basking spots among lizards, right? I'm sure there's similar things going on with amphibians, where in the wild they're competing for these spaces and these ideal microclimates. Yeah, so I think there's probably there could be some like genetic elements to this too. I think it's probably many things, some of those genetic or involved with their development of their phenotypes during development, and some of those related to ecology and interactions with other individuals, like landscape of fear stuff, like maybe you don't want to sit out on the warm rock all day because you'll get eaten by something. There's lots of reasons why individuals might choose the habitats that they choose. I think it's a really complex but really interesting idea. Yeah.
Thanks. We also have a question in the chat. In wild birds, avian influenza is often environmentally transmitted among migratory birds. Do you have any thoughts about how this hypothesis might extend to a system where hosts, and by extension their viruses, are seasonally migratory?
Yes. Let me see if I can. I might even have a slide, maybe not. But we did test migratory birds in that science paper, not migratory birds in particular, but we looked at differences between migratory and non-migratory species in their sensitivity to thermal mismatches. I'm just pulling it up so I don't quote this in my own paper wrong. But I could even potentially show the figure if I can pull that fast enough. Nope, I can't pull it fast enough. But yeah, it's certainly makes things more complicated. I think what we found was that there were like reduced effects of thermal mismatches on migratory species. And that is probably related to just being like the system being complicated, but also birds in particular like travel to make their climates ideal, right? They don't have to overwinter and experience winter stress. They don't have to hang out in the tropics in the summer and deal with heat stress then. They move. So they're buffering in some way. They're also endothermic, as I mentioned before. For endotherms, we don't see the same pattern. So they just have more ways to deal. They're also much more mobile than amphibians are. They have the ability to move around. So I think it's much more complicated when you think about systems like migratory birds compared to amphibians. It certainly makes it more difficult. I don't know that you would get that kind of pattern in avian influenza that we find with the amphibians. I would guess not.
Well, maybe we'll like pause there and Will can pause the recording in a second. But before that, let me just say that thank you, Erin, for coming. It's a great first talk. And next month, same bat time, same bat place is going to be, again, first Thursday at this time slot, is Lisa Cooper, if I'm getting that name correctly, and she does vector-borne climate interactions. So, and maybe with a little bit more of a human, health sort of flavor to it. So we're trying to move around. So as I said in the beginning, let's thank Erin, and those of you that are not students, you can go about the rest of your day. and go to your next whatever meeting that you have to do. But if you're a student, feel free to stay on, and we'll just have a brief discussion. If you have questions for Erin afterwards, feel free to stay on with us.